Good morning. So I'll be talking about cranial neural crest cells in craniofacial ontogenesis. So for in this seminar, we'll learn about I'll explain about the origin and migratory pathways of cranial neural crest cells, highlighting the molecular mechanism that regulate their epithelial to mesenchymal transition, which is EMT, and subsequent directed migration. Also, I will tell about the pivotal role of neural crest cells in craniofacial morphogenesis and emphasize on their contribution to skeletal connective tissues and neurogenic tissues. Also, I will discuss about anomalies associated with impaired neural crest cell functions, integrating embryological disruption with phenotypic outcomes and clinical syndromes. And lastly, I will summarize with recent articles in the field of neural crest cells in the past few years. So, neural crest cells, these are the cells which form in the embryonic stage of E7, around E7, and they can be divided on the basis of their location. The ones which form in the head region are the cranial neural crest cells, whereas the others are the trunk neural crest cells. These are formed by the folding of the ectoderm layer while during the formation of neural tube, and the cells which are present will migrate and will differentiate into various cells that can be melanocytes or neural cells or endocrine cells, etc. So neural tube and crest cell formation. So the neural crest cells arise during the neurulation stage, which in the early embryogenesis, and as the neural plate folds, it forms the neural tube, specialized border cells at the junction of the neural plate, and non-neural ectoderm undergo epithelial mesenchymal transition, enabling them to delaminate and migrate. And in craniofacial development, neural crest cells are particularly important because unlike the trunk neural crest cells, they retain the capacity to differentiate into ectomesenchymal derivatives, giving rise to much of the craniofacial skeleton. Then the pathways of cranial neural crest cell migration, we'll look into that. So after delamination, the cranial neural crest cells migrate in highly stereotyped streams, which is guided by gradients of morphogenesis and extracellular matrix interactions. These are distinct migratory streams and correlate with pharyngeal arch territories. So depending on the various arch, the cells will migrate and differentiate. So here are the few structures which are formed. As we know, the first pharyngeal arch will give rise to maxilla, mandible, and middle ear ossicles. The second pharyngeal arch will give rise to stapes, styloid process, and parts of hyoid bone. The third and fourth arch will form the lower hyoid, thyroid cartilage, and connective tissue. So all these will form from the neural crest cells migration. So cues such as efferent signaling, as well as the cell surface molecules such as semaphorins and integrins, they establish permissive and restrictive migratory corridors. So these expressions will allow the cells to migrate, and abnormalities in these signaling pathways will lead to craniofacial malformations. Then coming to how the differentiation and multipotency, we'll look into that factor of the cranial neural crest cells. So here is a brief table of how all these structures are formed by the neural crest cells. It forms the structure in the skin in the ectodermal derivatives. It will form the skin, nervous system, teeth, and eye. In mesodermal derivatives, it will form these bones and cartilage and fat tissues. And the ectodermal derivatives, it will form respiratory tract and thyroid gland and other structures. So cranial, they exhibit multipotency, and the lineage choices are governed by balance of intrinsic transcriptional programming and extrinsic cues from the surrounding environment. So it depends on both, which will form that is the ectodermal, endodermal, and mesodermal signal. So some skeletal derivatives, we'll talk about the structures which are related to the craniofacial area. So the skeletal derivatives, it will form the neurocranial, that is the frontal, temporal, parietal, occipital, ethmoidal, and sphenoidal bone of the skulls. And viscerocranium, that is the nasal bones, maxilla, mandible and other facial bones via intramembranous ossification. The cartilaginous elements will form that will be formed are Meckel's cartilage, Rachel's cartilage, laryngeal nasal capsule and lesser horns of hyoid, styloid process, mid and outer ear cartilage, sclera of the sclera cartilage or scleral cartilage of the eye. The connective tissue structures, it will form the dermis, tendons, ligaments, loose connective tissues and prevascular connective tissues of the face. And in the neurogenic derivatives, it will form the sensory cranial ganglia, Schwann cells, melanocytes, and parasympathetic ganglia. So, coming to the genetic regulation of neural crest cells. So, the neural crest cell development is governed by a gene regulatory network. The key transcription factors, these are SOX9, SOX10, FOXD3, snail, and twist. These are transcription factors. So they derive induction and EMT, that is ectomeson member transition, while signaling pathways such as WMT, BMP, FGF and Notch, so and Hox genes, so they will establish positional identity. Here are the various in detail. If you see all these genes and transcription factors and signaling. And then the epigenetic regulators, so the PRC12, E2H2 and chromatin remodelers and non-coding RNAs such as MRI302, fine-tuning lineage commitment. Then splicing factors such as SF3B4, it ensures correct RNA processing and survival of genes to regulate apoptosis via P53, and the disruption of any of these layers will lead to craniofacial anomalies such as Treacher-Collins syndrome and Deja syndrome. So we'll look out in these two syndromes in details. So these defects in neural cranial development, neural cranial crest cells development, migration, it will occur during the development, migration or differentiation and underlying several craniofacial syndromes. So these syndromes are illustrate how subtle disruptions in neural crest cells dynamics translate to complex craniofacial pathologies. The Treacher-Collins syndrome, it is also known as the mandibulofacial dysostosis. So the etiology, it is mutations in the TCOF1 pyloric or the pyloride gene, and impairing the ribosome biogenesis and inducing apoptosis of neural crest cells progenitors. So there are few clinical features which are seen. That is hypoplasia or of the zygomatic complex and mandible, downwards slanting of the palpebral fissures, that is on the border of the eye, and the ear anomalies will be seen, that is deformed outer ear will be there, and they are also associated with cleft palate. So the pathogenesis is the reduced cranial neural crest cells contribution to the first and the second pharyngeal arches will cause the Treacher-Collins syndrome. Also, there is parrot beak nose and underdeveloped mandible will also be there. There might be tooth agenesis and discoloration. Coming to the Deja syndrome, it is the deletion of 22 deletion in the 22 chromosome Q arm 11.2. So that is hemizygous deletion of genes affecting the pharyngeal arch patterning. So clinical features are the craniofacial dysmorphism, control cardiac anomalies, and thymic hypoplasia, immunodeficiency, and hypocalcemia. So the pathogenesis is the disruption of neural crest cells migration into pharyngeal arches 3, 4, impairing the derivatives of thymus, parathyroid glands, and the cardiac outflow tract. So here are the clinical features of Deja syndrome. It will affect the neurodevelopment, facial, the ENT also, endocrine, cardiac, immune, and others. So here are the few recent studies that are done in this cranial crest cells. The title post-transcriptional regulation in cranial crest cells expands developmental potential. So this study says that the MIR 302 family, it helps this gene, it will help in maintain or expand the developmental potential of cranial crest cells by slowing ectomesenchymal specification via targeting the SOX9 and repressing chromatin condensation genes. So loss of MIR 302 will lead to premature lineage bias and reduce neuron differentiation. Another study done titled the EED controls craniofacial osteoblast differentiation and mesenchymal proliferation from the neural crest cells. So this study shows the conditional deletion of EED, which is a part of the PCR2 complex after neural crest cells induction, will lead to severe craniofacial hypoplasia, impaired osteoblast differentiation, and reduced mesenchymal proliferation. Indicates that epigenetic control is crucial in the post-migratory neural crest cell gene program. Another study was done titled Etiology of Craniofacial and Cardiac Malformations in the mouse model of SF3B4 related syndromes. So the authors found that the mutation in the splicing factors of SF3B4 cause missplicing of critical cranial neural genes, including these genes, and this resulted in defects in the cartilage, bone differentiation, mandibular patterning, and shared craniofacial anomalies seen in humans SF3B4 related Nagar syndrome cases. So to summarize, the neural crest cells arise from the neural tube margins and migrate extensively to the craniofacial region. They differentiate into bone, cartilage, and connective tissues to form. They also dentine and making them central to the craniofacial morphogenesis. So precise regulation of their migration and fate ensures normal development, while defects will cause neurocristopathies. Syndromes such as Fletcher-Collins syndrome and DiGeorge syndrome exemplify the consequences of neural crest cells dysfunction. Thus, neural crest cells are vital for linking the embryonic induction events to the formation of complex craniofacial structures. So this for the topic. Thank you.